Welcome to Simplifying Synthesis, the channel where we look at complex organic chemistry and explain how it works. This week, we are going to look at some medicinal chemistry and see if DMT-inspired molecules could possibly rewire your brain and hold the cure for depression and anxiety. The work we are going to look at in this video was published by the Olson Group in the Journal of Medicinal Chemistry in a paper titled Identification of Psychoplastogenic NN-dimethylamino isotryptamine analogues through structure-activity relationship studies. So let's look at what psychoplastogens are. A psychoplastogen is a compound that can produce a measurable change in neuroplasticity within a short period of time following a single administration. Neuroplasticity is the ability of neural networks in the brain to change through growth and reorganization. This can range from individual neuron pathways making new connections to systemic adjustments like cortical remapping. Examples of these compounds include DMT, ketamine, and scopolamine. An important process in neuroplasticity is a change in dendritic branching, also known as dendritic arborization. Dendrites are the parts of a neuron which extend beyond the cell body and interact with other neurons. This is what forms neural connections. Complex dendrite branching is a feature of a healthy brain, and loss of arbor complexity in the prefrontal cortex is associated with conditions such as depression and anxiety, and is often triggered by stress. It is the pyramidal neurons in the prefrontal cortex, which control areas of the brain responsible for motivation, fear, and reward. Psychoplastogen-promoted changes in these neurons provides a potential explanation for the known antidepressant, anxiolytic, and anti-addictive effects of psychedelics such as DMT. Psychoplastogens related to DMT include psilocin, which is usually found in magic mushrooms, and lysergic acid diethylamide, more commonly known as LSD. These compounds contain a common molecular structure with DMT, which allows them to interact with brain receptors in a similar manner, which we will examine in detail throughout this video. These compounds are thought to promote neural growth by acting on receptors which trigger cell proliferation. These include the AMPA receptor, which is a type of glutamate receptor, and is named after the ligand alpha-amino 3-hydroxy-5-methyl-4-isoxazole propionic acid, a molecule known to activate it. The TrkB receptor, short for tropomyosin receptor kinase B, is also thought to be involved, in addition to mTOR, which is the mammalian target of rapamycin. Upon activation, this forms two complexes, known as mTORC1 and mTORC2, which are able to promote neuron proliferation. So let's start by looking at the structure-activity relationship studies. These studies are one of the primary ways in which medicinal chemists study molecules and optimize new drugs. It involves modifying the chemical structure of a compound and seeing what impact this modification has on the biological activities of the molecule. So let's look at the compound design. DMT is quite a simple molecule. It consists of an indole ring substituted at the 3 position with an ethyl chain bearing a dimethylamine group on the terminal carbon. This study investigated isomers of DMT, which placed the alkyl group on the nitrogen instead of the carbon at the 3 position to generate an isomer, hence the iso in the name of these compounds. This change improved several key parameters, which must be optimized during drug development. These include pKa, which is a measure of how acidic or basic a compound is, C log D, which is the calculated distribution coefficient. This is similar to the more common partition coefficient and is used for ionizable groups such as amines. It measures how lipophilic a compound is and is related to how well a compound will be able to permeate into a cell. Another factor is the topological polar surface area, which is the surface sum of all polar atoms in the molecule. and the number of hydrogen bond donors. For drugs acting within the brain, the multi-parameter optimization score can be used to assess the likelihood that a compound will be a successful drug. This takes into account several factors which affect the pharmacokinetic and toxicological properties of a compound. Looking at the predicted values for these two compounds, we can see that the iso-DMT compound is predicted to be a better drug candidate and more effective at permeating into the brain, which is one of the main challenges in designing CNS active drugs. So let's look at the first experiment, which was to probe the effect of isomerization. This study was designed to see if swapping the alkyl group between the carbon and nitrogen would affect the psychoplastogenic activity, and also see if the NH bond is necessary for activity. To do this, they would assess the effect of DMT, 1-methyl-DMT, and iso-DMT on dendritic arbor complexity. The methods for this experiment were to visualize the neurons using an anti-MAP2 antibody and determine the dendritic arbor complexity using Schall analysis to count dendrite crossings. MAP2, or microtubule associated protein 2, is a cycloskeletal protein localized in the somatodendritic compartment of neurons. Using an antibody which binds specifically to this protein allows scientists to visualize where this protein is by using a fluorescent label which can bind to the anti-MAP antibody. This allows dendrites to be visualized. So here we can see the images of the neurons which were grown in the presence of these compounds. The first image that we can see here are the neurons that were used as a control. These were not grown with any drugs present, and instead just the vehicle which was used to administer the drugs, which was a serum-based buffer. As is clearly visible, the neurons that were treated with DMT, 1-methyl-DMT, and iso-DMT all show an increase in the dendrite complexity. The graphs of the Schall analysis show that all of these compounds cause an increase in arbor complexity, indicating that they are working as psychoplastogens. We can conclude, therefore, that the alkylation of the nitrogen atom and the loss of the NH hydrogen bond donor does not affect the ability of these compounds to promote neuroplasticity. So now let's look at the next experiment, which was designed to look at the influence of modifying the aromatic rings present in the molecules. To do this, they synthesized a number of analogues and varied the number and nature of the aromatic rings present in the molecular structure. They then treated neurons with these compounds and visualized them using the same methods as we saw earlier. From this study, we can see that all of the compounds they tested retained their ability to act as psychoplastogens. This included compounds with an extra aromatic ring, or even with just one, in addition to compounds with an extra nitrogen atom and electron withdrawing group, which changes the electron density on the aromatic ring. This experiment also utilized ketamine as a positive control. This was used as a benchmark to compare the novel compounds against, as ketamine is one of the strongest psychoplastogens known. The next experiment involved looking at the basicity of the nitrogen atom. To study this, they synthesized two analogues, one without a nitrogen atom, and two with the nitrogen atom, but with the lone pair delocalized with a carbonyl group, which reduces its basicity. The effects on arbor complexity were quantified using the same Schall analysis as we saw before. The results of this study show that the basic nitrogen is essential for activity, as the two analogues synthesized showed greatly reduced activity when compared to DMT and ketamine. In the final study carried out to define the Pharmacoform, the authors studied the influence of the substituent position. For this experiment, the authors synthesized analogues with a methoxy group, which is electron donating, a fluoro group, which is electron withdrawing, and a benzyl group, which is electron donating and sterically demanding. They then studied the psychoplastogenic activity of these molecules. From these results, we can see that the substitution was well tolerated at all positions, with the exception of the 4 position. For all of the molecules studied, when the 4 position was substituted, a reduced activity was observed. As there are no crystal structures of DMT bound to the 5-HT receptor, it is difficult to say with certainty why this position is so sensitive to substitution. However, the crystal structure of LSD bound to the 5-HT2B receptor has been published, and examining this suggests that the area occupied by substituents at the 4 position is occupied by helix 5 of this receptor, therefore blocking any molecules which are substituted in this position. This is still a hypothesis and is yet to be proven. So with the pharmacophore now defined, the authors looked at the role of the 5-HT2A receptor. This receptor is a G protein coupled receptor, and its natural ligand is serotonin. This receptor is known to bind psychedelic compounds such as DMT and LSD. To do this, they would assess the effect of iso-DMTs on dendritic arbor complexity in the presence of a 5-HT2A antagonist. An antagonist is a molecule which can bind to a receptor without activating it. By binding to the receptor, it will block it and prevent the test compounds from binding. In this study, they use ketanserin, which is a known antagonist of the 5-HT2A receptor. By administering the compounds with and without the antagonist added, and then visualizing them, the scientists can assess if these compounds are binding to the active site of this receptor. From these studies, we can see that this receptor is essential for the psychoplastogen activity of these compounds. In the samples where ketanserin was present, indicated with a plus sign in the graph, we can see that the dendrite growth was greatly inhibited and showed a result similar to the control sample. However, in the absence of this antagonist, these compounds show activity similar to that of ketamine. So now let's look at the animal studies which were conducted. In the first experiment we will look at, the authors sought to examine the behavioural bioisosterism. This was to determine if the DMT and iso-DMT molecules cause the same effects on behaviour. To do this, they would administer these compounds to zebrafish larvae and analyse their behaviour in response to stimuli. The method of this experiment was to stimulate the larvae with light, sound, and touch, and then monitor how much they move. They would then train a multi-classification model to distinguish between sample populations and assess out-of-the-bag accuracy of random forest models. To put this simply, they would use machine learning to train a model to distinguish between larvae which had been treated with different compounds, and to see if it could distinguish between larvae which had been treated with DMT or iso-DMT compounds. So here we can see the results of this zebrafish study. In the traces on the top, we can see the locomotion, which is how much the larvae move. Along the bottom, we have the stimuli. The black bars correspond to acoustic stimulation, the coloured bands correspond to light stimulation and the wavelength of the light that was used. And the grey bands towards the end of the study show the response to touch, the light grey corresponding to a light tapping, while the darker grey is a more forceful tapping. All of the compounds which were studied induced a change in the behaviour of the zebrafish larvae. These include a reduced response to acoustic stimuli and an increased response to blue light. In these graphs, we can see how accurate the machine learning model was at distinguishing between larvae populations which had been administered with the test compounds and those which had not. The confusion matrix shown on the right showed that misclassification of isosteric pairs, that is, related DMT and iso-DMT molecules, was more likely and indicates that isosteric compounds show more similar behavioural effects in the zebrafish larvae. So in the final experiment of this study, the authors looked at the hallucinogenic potential of these compounds, which is, do these iso-DMT compounds cause hallucinations in the same way that the psychedelic DMT molecules do? To do this, they would administer DMT and iso-DMT molecules to mice and analyse their behaviour. The method they would use to quantify the hallucinogenic potential was head twitch response, which has been used previously as a proxy to determine the hallucinogenic effects of molecules. As we can see from these results, 5-methoxy-DMT produces a strong head twitch response, indicative of hallucinogenic potential, while its isomer, compound 6, induces a much weaker head twitch response, suggesting that it is a weaker hallucinogen. Meanwhile, the 6-methoxy-DMT, compound 30, did not induce a head twitch response, which is in agreement with previous studies on this compound. Compound 5, the iso-DMT, also did not induce a head twitch response in any of the mice studied, suggesting that it does not cause hallucinations. This would indicate that compound 5 shows great potential for further study as a psychoplastogen with medicinal uses, as it does not cause hallucinations, but has been shown to act as a psychoplastogen. This could suggest that it may have uses in treating conditions such as depression and anxiety. While it is promising, a lot more research would need to be done before this could be a viable drug candidate. So finally, let's look at the synthesis of these iso-DMTs. The great advantage of isodimethyltryptamines compared to regular tryptamines is the ease of synthesis. To make these molecules, you simply react the substituted indole with a chloroethylamine with base, in this case potassium hydroxide, and a catalyst, potassium iodide, which acts as a nucleophilic catalyst. This alkylates the nucleophilic nitrogen atom of the indole group. These compounds can then be salted out with fumaric acid and recrystallized without the need for any chromatography. So to summarize this work, the authors have shown that iso-DMT molecules show similar psychoplastogen activity to the psychedelic DMT family of compounds. They likely act through a similar mechanistic pathway mediated by the 5-HT2C receptor, and they show low hallucinogenic potential, suggesting that they don't cause the same psychedelic effects as DMT. In addition, these molecules are very easy to synthesize, which means it will be easy to explore this family of compounds further. That's everything from this week's Simplifying Synthesis. If you enjoy these videos, please like and subscribe, and if you want to see more medicinal chemistry, please let me know in the comments down below. I'll be back next week, where we will look at the total synthesis of cinnamodial-based dimer capsicodendrin.